before we start the seminar, following our tradition, just to announce that Horacio Bio, which many of you know, is now with us. He's as an associate researcher with the first appointment of 3 years. So welcome, Horacio. He's on the first floor. I'm sure happy to interact with many of you.
Thanks, Maxi. And I would also like to welcome two shorter-term guests we have, both Konstantin Blyuss and Julia Kilichko. They're both senior lecturers in mathematics from the University of Sussex. And they will be with us for the next 2 weeks. And coincidentally, they are also the two next seminar speakers. So the start will be made by Konstantin Blyuss. Kostya has a quite interesting CV from the complex systems point of view. He got actually 2 masters in physics, one from Dniepropetrovsk, I hope I pronounced it correctly.
It's one of the best I've heard over the years.
Trying to smoothen it. And also in theoretical physics from the University of Brandenburg, correct? Brandenburg Technical University. And in 2003 he got his PhD in mathematics then from the University of Surrey in the UK. And the title was Perturbed Multisymplectic Systems, Intersections of Invariant Manifolds and Transversal Stability. So you see already, he's already back then working on complex systems. He became from 2003 to 2006 an EPSRC Research Fellow at the University of Exeter. Then went to the University of Oxford as a postdoctoral mathematical biologist. From 2006 to 2008 he was a temporary lecturer in applied mathematics in Kiel University in the UK. And from 2008 to 2010, that's where we met first, I guess. He was lecturer in complexity science in the Department of Engineering Math in the University of Bristol in the UK. So he stayed faithful to the UK and moved in 2010 to Sussex, University of Sussex, where he's a senior lecturer in mathematics. And since 2015 he's also a fellow of the Higher Education Academy in the UK. So his research topics he's interested in is mathematical epidemiology and immunology. Oh, see, I start to stumble. Delay differential equations, in particular role of delays in oscillator networks, gene regulatory networks, neural systems, immune response, network dynamics, and applied dynamical systems. And today he's going to tell us something about mathematics of RNA interference. Of course, we're very happy to have you here, and thanks. The floor is yours.
Thank you so much, Inga. First of all, thanks a lot for a really wonderful opportunity to give a talk, but even more importantly for absolutely amazing opportunity just to be here. And again, the weather you've arranged, well, there are no words to describe it, because when we were leaving the UK it was rainy, +7 and quite miserable. So again, coming to an institution and looking outside, again, the first thing, and I'm not lying, the first thing I saw to Inga when we saw him on Monday was, what bugs me is I can't understand how you can get any work done here. So you do have my full sympathy. If you want any proper work done, come to the UK. So what I want to show you today is something that, again, it's not really too mathematical, but it's more how we can use mathematical models to understand a really important biological process. That's what the whole thing is about. And it's joint work with Julia, who will give her own talk on a completely different topic in a week's time, and also Giannis Neofito, who was our joint PhD student, and he just completed his PhD about a month ago. So what I'll tell you is, first of all, I'll explain what the RNA interference is, why we care about it, where it shows up, and why it is actually a very important thing to understand the mechanism of. And I will focus on 2 different parts of it, or 2 different aspect areas, if you like. So the first one is RNA interference in plants. I will explain why it's important, why we care about it, and also how mathematical models can provide some intuition and some understanding of the dynamics of RNA interference in plants. And then we will talk more specifically about actually what happens inside the cell, so how that actual RNA interference as a process works. And in both of those cases, I will focus on the role of time delays. which again, sometimes one could argue that you can get away without explicitly introducing them, but here, again, there are absolutely valid biological reasons why these things should be included. And I will finish with some conclusions and open problems. And if there are any questions during the talk, don't be shy, just ask. So, what's RNA interference? That's the picture from the Nobel Prize website, because, as I will tell you in a minute, 2 guys got a Nobel Prize for it. The idea is the following. So, in every cell, what we have? We have DNA, which then is transcribed into mRNA. mRNA is translated into proteins, and proteins play all sorts of important roles. If we didn't have them, none of us would be here today. Now, what is RNA interference? This is a really fascinating mechanism which effectively can stop proteins from being produced. How does it work? So it starts by having what's known as a double-stranded DNA, which by a particular enzyme is cut or spliced into short pieces. They're still double-stranded. Then they are unwound, and one gets 2 pieces of single-stranded RNA. That then becomes mRNA, and what we have in here is what's known as RNA-induced silencing complex. So out of those single-stranded RNAs, or short interfering RNAs, inside the cell, there is a possibility to construct something that floats around and looks for homologous mRNA. And the moment it recognizes it, it degrades it. So if you have a virus or transposon or anything else that tries to increase its own presence inside the cell, it only can do it by increasing the amount of mRNA that would then be translated into protein. And whilst cells cannot stop the initial stage of the process, so DNA is still transcribed into mRNA, they can arrest it at the next stage. So they can, for viruses for instance, they can stop them being realized in terms of proteins, because this last stage of translation from mRNA to proteins just doesn't happen. Why? Because there is this RNA-induced silencing complex that floats about and just degrades all of that unnecessary mRNA. It happens in all kinds of cells. So the very first examples were in plants, but then it was discovered everywhere. The really important process, or rather the really important role of this process, is effectively to regulate expression of individual genes. Because in the DNA you can have all kinds of genes, they all have different roles. Some of them are important for particular processes. It may be development, it may be immune responses, it may be all kinds of things. But the point here is that RNA interference is a mechanism or a tool that allows you to effectively stop or arrest expression of individual genes. And in the particular example of plants, it's effectively the first line of viral defense, because plants, unlike mammals, they don't have what we interpret as the adaptive immune system. So we have evolved over millions of years to have a very robust immune system, thank God. But with plants, it's a different story, so they operate differently. But still, there is nothing that prevents them from being infected by viruses, and still they have to respond somehow. And RNA interference is actually a mechanism by which they're able to stop viruses that can come inside those plant cells from reaching the stage where they produce viable proteins. And also, this is a very convenient and very effective experimental technique. Why? Because it provides you with a very targeted and quite accurate mechanism by which, if you have to knock out a specific gene, or if you want to ensure that no protein will be produced, will be effectively expressed from the gene, that's how you would be able to do it. Because these RNA silencing complexes, they are very gene-specific. So they would only recognize a particular RNA sequence. So perhaps the first paper where this phenomenon was discovered, again, they found it but did not really understand at the time what was behind it. It's more that they made an experiment, they had an observation which they reported. But it was a breakthrough because it went against all intuition. So what these people did was they looked at petunias and they wanted to increase the amount of blue color in them. So they overexpressed a particular gene, and they expected to get something that would be looking like this. In the end, they ended up with petunia that was completely white, which just didn't make any sense, because they looked at the gene that, for all intents and purposes, everyone knew was responsible for the blue color. They increased the expression of the gene, so surely they would expect to have more blue color, so they expected the plants to be completely blue. And they weren't. Rather, the exact opposite happened, so that gene in the end was knocked out. Same was observed in a completely unrelated study, and they didn't know about each other. At the time, that process was called quelling. These are all names for exactly the same thing. It's just that since they were discovered by different groups in completely different contexts and at different points in time, it took a long while to realize that actually this is all one and the same biological process. So quelling is what happens in mold, and C. elegans, so that's a nematode, and this was the one that got the Nobel Prize, because this was the one on experiments with which it was finally understood what the mechanism is and how it works. And there are different constituent parts of this process that I'll walk you through that explain actually what is happening. So this was the paper that got the Nobel Prize because at the time no one understood what is the starting point or what is the origin of the RNA interference, so what really triggers it. And those two chaps, they figured out that actually it's the double-stranded RNA that is the first necessary ingredient to get the whole process going. So later on, of course, once people understood that what the mechanics of this is, it was discovered in all kinds of species, from fish to mammals to lots of parasites, but again, in many parasites, but not in all parasites, because again, an organism must have quite developed genetic machinery in order to be able to work with RNA interference. So, if we look at RNA interference in plants, first of all, why do we care about it? Is because of plant viruses. So, the overall world population is growing, the demands for agriculture are growing tremendously, and that's a rough estimate of the financial losses only to just purely due to losses of plants to viruses and other pathogens. Obviously, it's not just the financial losses, because there are lots of communities worldwide that rely almost solely on the agriculture for sustaining themselves. So if there are any problems with crops, it can have also huge societal challenges. So the idea is we want to understand how plants respond to viral infections, and if we understand that, then maybe we could come up with better ways to overcome those plant infections and thereby reduce the problems with pathogens in plants. So here's a simple model. What's the idea? We take a plant. A plant is growing. So there are some cells that are just proliferating, the new cells. When they're new, and that's what the experimental evidence suggests, they actually cannot get infected. So if new virus particles come about, then they can only affect the cells that are already mature. So at the initial stage, when effectively the plant is just growing, be it leaves or the stem, proliferating cells, they are completely immune to infection. At the same time, they also lack the machinery to actually facilitate RNA interference. So in that respect, they are, if you like, in their own protected class. So they don't fall vulnerable to infection, but also because they don't need to, they don't have RNA interference inside. After a period of time, they mature. That's when they start to play the game. So at this point, they can be vulnerable to an incoming infection, and they can develop RNA interference that would allow them to overcome that infection. Obviously, they start by being mature and susceptible, and then once they become infected, that's when they are in the class of infected. And in the infected class, what can happen is actually infected cells can recover. And again, this is a really interesting thing, that it's not that cells can only die. In this case, and again, it's just an experimental observation, cells actually can clear the infection. They clear the infection because they can use this RNA-induced silencing complex to effectively completely arrest the production of any new virus particle from inside the cell. So effectively, a cell becomes immune. And then there are born cells. What are those? These are parts of the plant that are already mature. So in principle, they could become infected. But because they have received some warning signal from other cells that have already been infected, now inside them, they start producing this RNA interference at large quantities, so that actually it makes them completely immune. So they cannot be infected. Because even if there is a viral challenge, so if virus particles try to get inside, they will be immediately destroyed just because there is this RNA interference inside. Now, how do we formalize this? So these are the cell compartments that we look at. At this point, there is no space. So even though, of course, every plant leaf has a geometry and it's growing and things are happening to it, for the purposes of this model, we assume that things happen homogeneously in space. But in a second, I'll explain what we still do take into account. So we have P is the proliferating cells. So these are the ones that effectively are just born. They cannot become infected. They have no RNA interference. After a period of time, tau 1, they become mature cells. So in the class S, S stands for susceptible. They can become infected, at which point they would move here. But also some of these cells can become warned because they can receive warning signals from other cells. To go from here to here, we have this factor. The interpretation of this is these are all cells that have survived for a period of time tau 1 from the natural death rate. So epsilon is natural death rate. So these are all those of those growing cells that effectively have survived to maturity, at which point they go in here. They can become infected. From infected, they can recover just by natural spontaneous recovery, or because they have also received a warning signal, because warning signal brings with it RNA interference. So that's the mechanism that internally allows infected cells to clear the infection. So that's another contribution of them going to the class of recovered. And since infected cells, effectively all their efforts go into fighting infection, it means that they and also the recovered cells, because they have experienced such a major challenge, none of them can contribute to the new cell growth. So it's only the susceptible cells that do not have infection and the born cells that are even in an even better position because they have not had any infection and cannot get any. It's only those two classes that contribute with rate K to the production of new proliferating cells. So that's what we have. Any question? So here's the model. In here, I have only 3 compartments because effectively there was another one for proliferating cells, but we can get rid of it. It's just this term. What do we have? The number of susceptible cells. So K times S of T minus tau 1 plus W of T minus tau 1. These are all the cells that were born time tau 1 ago. That's how many of them have survived from natural death. Minus everyone who is here can become infected, at which point they would go to the class of infected. They can also receive the warning signal, and all of those would go to the class of born cells. And this term, effectively, it's like the logistic growth, the term that limits the unbounded growth. So that if there are no any other classes, we still don't want to have unbounded growth of the susceptible cells. In the infected class, we have natural death, which has obviously happens at a higher rate than for susceptible cells. We have the recovery, and we also have the warning signal, because that's so where does the warning come from? If a cell becomes infected, it starts to fight the infection, and as a product of that, it also produces this double-stranded RNA or mRNA that can travel to nearby cells And it's this warning signal that goes to nearby cells, and when it arrives there, it either turns them into warned cells if they were previously susceptible, or if it arrives to a cell that is already infected, it will help it recover faster. And tau2 is effectively our proxy for how long it takes for a cell from the moment it becomes infected to when other cells start receiving signal from it. So it takes into account obviously several stages, from the actual production of the warning signal to the time it takes to spread. Biologically, there is every good reason that this term is there. The really interesting question from a physics point of view is, well, can we measure it? And unfortunately, at this point in time, the answer is we would love to, but we do not know how. Why? Because among the plant biologists and more generally about biologists, everyone agrees, because all the evidence unequivocally suggests that yes, there are these warning signals that are spreading. So that's without question. There is evidence for that because infection happens at one part of the plant, but warning signal is definitely affecting what happens in other parts of the plant. If you ask the question, well, what is the actual signal? So is it mRNA? Is it double-stranded RNA? So what is that signal that is actually spreading and at what speed is spreading? The answer to that, no one really knows. So because depending on individual setups, either mRNA was found long away geometrically from where it originated, so clearly it is spreading. In other experiments, it was the double-stranded RNA that was observed in other parts of the plant. So it looks like there are various mechanisms that effectively contribute to this overall role of the warning signal. And we have two time delays. So tau2, exactly because we can't at this point of experimental science quantify which of the signals is exactly spreading. We know they're spreading. We take tau2 to be a proxy for how long it takes from the production of RNA signal to reach susceptible or infected cells. For susceptibles, it would turn them into warned. For infected cells, it would just help them to recover. And, right, so besides not terribly interesting result that the model is proposed and everything's fine, yeah.
I have one question, if you can go one back. Regards mostly what you call the kind of logistic term that you have first. First, why the two contributions? Because this would be S, I think, instantaneous time times S, I think instantaneous time. Why not one of them is delayed? I mean, in this logistic limitation, in principle, I mean, you would have some delay. I mean, the standard logistic equation with delay normally, I mean, has delay in this.
No.
No?
No, stop. In the normal logistic model, the simplest one you would take, it's dm by dt is a constant times n times 1 minus n divided by carrying capacity.
Yeah, sure.
So you wouldn't have time delay there. You wouldn't want to...
Extensions of
you could.
Equation with delay.
In principle, you could do that. But then, again, it wouldn't make any sense. Why? Because the whole point of having time delay there is if you have a population of cells or any biological species where experiments suggest that it has periodic oscillations, then having time delay in the logistic model is a cheap and easy way to get there, because then you would just have very large oscillations around carrying capacity. But here, because we are talking about just the plant cells, there is no reason to say that if there is no infection, no warning signals, nothing, then effectively the plant is just growing. And ultimately we say that a leaf is growing until it reaches some mature stage. So for any plant, when we go outside, leaves are of some average size. It's not that during time evolution they grow and shrink. So that just wouldn't be feasible. And this is purely just a reflection of how many susceptible cells in the plant or in the leaf we have. Which is why at this point we didn't think it would make sense to have an extra time delay there.
We have another question with this term. Epsilon, I guess that it's the death rate of the worn cells. Why is also related to the current capacity? I mean, 1 over...
it's more that...
1 over epsilon or something like that.
Well, effectively it's, well, minus epsilon s squared is the death term here in the first equation. So in that respect, it is a version of a death rate. I agree that it also has, in terms of dimensional units, part that's related just to the number of cells, but again, in principle, there is nothing that would stop us from having this as any other constant that would not be epsilon.
And why don't you use the same kind of mechanism with the worn cells? Because no, they grow in the same way as the
No, they don't grow in the same way.
Okay.
The major distinction is that, well, if so born cells, they can only appear out of susceptible cells.
Mm-hmm.
But if there are no born cells, then there would still be susceptible cells in here and in here. So say, you could ask the question, why did we have this term as epsilon s squared, whilst for all other terms we just take minus z times I or minus epsilon times W. So in all other equations, the death term is just constant times that population. And the answer is that for any of the other populations, it wouldn't be a problem. But for the very first one, it would be. Because if there is no infection and there is no warning, then if we just have this term and we just have a constant times s, then we would either be exponentially growing or exponentially decaying. And that's what we wanted to avoid. So for the very first one, we wanted to ensure that if there is no infection, it still stays bounded. And for the other ones, it wouldn't be a problem. Because for all the other ones, because won't only come from S, which is now bounded, so it cannot grow as much as, well, it cannot grow unbounded. So that's the reason.
Is the origin of the nonlinear term in the infected, the delta phi, this nonlinear term, what does?
This one?
Yes, yes.
Right, let's talk about this. So in here, I of t minus tau 2, that's a proxy for for some warning signal that originates inside the infected cell and then started spreading. Then as it's spreading, so delta times this term, that's effectively, if you like, a proxy for amount of the warning signal that reaches susceptible cells. Now, the reason why we have here an extra phi is for the following reason. These are infected cells that also have received the warning signal delta times I, but then it doesn't mean that that's exactly the number of those infected cells that will therefore recover. So in both of those cases, delta times I, that's the warning signal, if you like. And this effectively is a conversion factor.
So this means that if you are infected and you receive warning signals somehow,
yes,
you recover faster.
Yes,
As
absolutely. So here there is this term which is just normal recovery if there is no warning signal at all, and then this is an extra recovery that's associated.
Recovery if you receive signals
absolutely.
from other cells which are infected,
absolutely.
Then you produce more
absolutely right. Yes.
substance or whatever.
Yes, absolutely. Yeah. So this is the spontaneous recovery if there is no warning, and this is an extra warning signal that has arrived, and if you like, phi is a conversion factor that tells you well,
how much is the effective
yeah,
of that
exactly.
Yeah.
But obviously both of them are with minuses, so the respective terms would go to the recovered compartment, which again, the reason I didn't write it here is because none of the other variables depend on it explicitly. So we have a trivial steady state where there is nothing. So no susceptible, no infected, no warm cells, and thankfully it's always unstable. So that's how we wanted it. It's unstable irrespective of the values of time delays. Then there is a disease-free state, which does depend on the maturation time. And again, here we can find conditions on the maturation time delay so that the state would be itself stable or unstable. And then we have the most interesting one, the endemic state. So that's where all the compartments are positive. We can find explicit expressions for all those components. Again, this steady state now is not always feasible. So it's only feasible if the maturation time delay lies in a certain range. It's effectively the same thing as with standard infections or standard epidemic models, in that if you want to have an endemic steady state, then transmission rate should effectively be faster than the recovery rate. Because if infection is cleared very quickly, then there is no way how it can be stably maintained in the population. Somewhat similar logic applies here. Now, if we look at the stability of this steady state, what is the problem? So we have our 3 compartments, but we also have 2 time delays in the system. So when we look at the characteristic equation and say that tau1 is equal to 0, which physically means that cells mature immediately. So from the moment they're produced, they straight away go to the class of susceptibles. Then we get a cubic equation, but it's not really cubic because it also has these transcendental terms. So tau2, remember, is the time for the spread of the warning signal. But still, we can do actually quite a bit of analysis on this, because we can start by saying, suppose the second time delay is also 0, and then we just get standard Rauch-Hurwitz conditions for stability. And then when we increase time delay, we can find what is the possibility for the Hopf bifurcation, because this endemic steady state, as the time delay is increased, a pair of eigenvalues can cross the imaginary axis, and when that happens, we would get a critical time delay that tells us so that if the time delay is above this, then the endemic state would be unstable, and we could expect to see periodic oscillations. You could do the exact opposite. You could say, suppose that the warning signal is propagating very fast. So fast that it would be smaller than all the characteristic timescales in the system, that effectively we can set the time delay for the propagation of signal to 0. And then, again, if we still keep the maturation time delay, we would get a very similar kind of mathematical challenge. Again, we can analytically find the critical value of time delay, we can find the Hopf frequency, which would then give us the estimate of the period of oscillations. Another possible reduction is, again, well, that's just a mathematical trick. The purpose of this is purely to go from a system with 2 time delays to 1 time delay. Because with 1 time delay, we can do the full analysis. With 2, unfortunately, we can't really. So if, for argument's sake, we say that the 2 time delays are about the same, then again, the problem reduces to a cubic equation, but the major difference between this and everything I showed you before is that, unfortunately, here, the coefficients of the equation, they themselves depend on the time delay, which hugely complicates the analysis. Therefore, we couldn't do anything more analytically, so we had to do numerical bifurcation analysis. We started with some estimates on the parameters. Again, with this, I have to put my hands up and say that these are completely arbitrary. I cannot tell you what units they're in, whether time is measured in seconds or hours or years. Why? Because we just do not know. And unfortunately, in biological literature, there is such a vast difference in reported values for lots of them that whilst the mechanism everyone agrees on, but the actual measurements are very far from us to be able to use them in simulations. What do we have? So that's the maturation time, and that's the time for the propagation of the warning signal. So K, that's the rate at which the new cells are produced. So if they're produced not too fast, then if the maturation period is sufficiently high, then we just have the stable disease-free steady state. That's the blue part. Below, that's so the color shows the largest real part of the eigenvalue associated with the endemic state. So whenever it's green or yellow, that's where it's unstable. Where it's purple or blue, that's where it's stable. And this is really interesting and quite counterintuitive. Why? Because if the warning signal propagates very fast, so that's what it means to have small tau2, then actually the endemic state, so that's the one where infection is persistent, is stable. So that's what this simulation suggests. And qualitatively, this picture persists if we go to a state where we increase the rate of producing of new cells. The major qualitative distinction between those two figures is that here the endemic state exists everywhere in this region, whilst here at the bottom region the endemic state just doesn't exist. So only the disease-free exists but is unstable. So disease-free is stable here and unstable everywhere below. And in this case, the endemic state, so that's the one where the infection is persistent, it only exists in this bounded region. And again, it's stable for small values of the propagation delay and unstable for large values of propagation delay. Again, we can look at how different factors affect stability, such as, for example, infection rate. So if the infection rate is very fast, then it doesn't really matter what is the speed of the propagation delay, because the dynamic state will just always be stable. And again, what this picture also shows that in order to have a stable and sustained persistent infection, you do require some minimum level of the transmission rate. Because if you don't, then effectively infection is cleared faster than the new infection is produced. And again, we just found all the different kinds of regions where either disease-free state exists always, and the endemic can either exist or not exist, or be stable or unstable. And this is what it looks like in terms of simulations. So these three figures, the blue color, that's the susceptible cells, red are infected, and black are warned. So in these three cases, we have what in biological interpretation would be a chronic infection. So eventually the system settles on a stable state in which all three populations are different from zero, so infection just stays in population forever. This is what biologically could be interpreted as a recovery phenotype. So there is some initial infection that grows, but that's the red curve. But then it's cleared, and that's it. So the system goes to a stable steady state where there's only the population of susceptible cells. Warned also disappear because there is no more warning signal. It can only come from infected cells. So effectively, that's just normal recovery. There is initial viral challenge. The number of infected cells goes up. Then infection is cleared, and that's it. And in those two, these are endogenous oscillations, purely because of the time delays that we have in the system. So here, it's not that we need to have some periodic challenge to the system to ensure that we have this. It's more that the intrinsic dynamics of the system is such that it not only permits, but also sustains those periodic oscillations, obviously around the endemic steady state. So the state is unstable, but around it there is a stable periodic solution, and that's what we see. Any questions so far? If not, then let's completely change tack. So everything I have shown you so far, it was at the level of population of cells. So we had plant, and we had body signal that was spreading from some cells to other cells. Here, everything in this story is happening within a single cell. And what are the constituent parts that we care about? So mRNA, that's something that is produced inside the cell, and that's something that can be detrimental to the cell. And the point is, RNA interference tries to get rid of that mRNA because it is perceived as harmful or unnecessary. So there is double-stranded RNA that can just get produced at some rate from this mRNA, and then part of it is sliced by the DAS enzyme. It becomes a short interferon RNA. So this was something that was found in the very early experiments, and people thought, right, short interferon RNA, that's what causes the RNA interference. Until it became clear that in some cases, when people didn't have this, they could still have RNA interference. So it must have been something else. And it's the discovery of this double-stranded RNA for which Nobel Prize was awarded. Now, short interferon RNA, it's produced from chopping up of the double-stranded RNA, and N is the number of copies you acquire. And then this BSM, so what happens here is there is this term, short interfering RNA gets into that complex which I called, or rather they called RNA induced silencing complex, which floats about and destroys mRNA. That's the rate at which this process happens. So logically, we could have a separate compartment that would describe just that silencing complex, but that would make the system much larger, and there is no point in doing that. Rather, we take the amount of short interfering RNA as a proxy for how much of those silencing complexes there would be about. And then BSM, that's the effect of that silencing complex. Obviously, because it gets bound to that mRNA, it also reduces the amount of free short interfering RNA that's still available. And at this point, this term goes to here, where G stands for garbage RNA. So these are all the compartments in the system. And in this paper, which now is quite old, they looked at different versions or different aspects of how this can be modified. Either that the short interfering RNA can be degraded at some additional rate, or that there may be additional production of double-stranded RNA directly from the garbage. So effectively the cell takes the short interfering RNA as a template and produces an extra double-stranded RNA that's helpful from this garbage, which is this term. Or that the double-stranded RNA could be produced directly from garbage at this rate, and then it would go here. Now, the reality and the reason for what we did next was that it actually takes some time for those processes to take place. And we took this model and we looked at two particular effects or two particular processes. So in here we have the same compartments, but we also have two time delays. What do they represent? Let's have a look. So tau 1, which is this one. What do we have? So B1 SM, that's the standard destruction of mRNA by the RNA silencing complex. B2, that's the production of new double-stranded RNA from mRNA and the short interfering RNA. But when they get together, it's not that the very same moment you have the double-stranded RNA. That process actually takes some time. So whilst they disappear from those two compartments, they do not immediately reappear in this compartment. So the interpretation of tau 2 is this is the amount of time that is required for production of double-stranded RNA from the mRNA itself and the short interfering RNA. Likewise, this term. So we have mRNA and short interfering RNA. They get together, and this is the amount of time that is required to produce double-stranded RNA from those two. So in both cases, it's the amount of time that it takes to produce double-stranded RNA from short interfering RNA and something else. In the case of tau 1, that something else is just mRNA itself. In the case of tau 2, that something else is garbage RNA. Again, there is good biological reasons for doing that. There are also estimates on some parameters, so at least here we are on a slightly safer ground than before. What happens when we look at steady states? So here, unfortunately, it's more complicated because the system is much more involved and we have 4 variables. So when we look at the amount of short interfering RNA, it satisfies a cubic equation. But because of what the coefficients are, we know for a fact that there will always be at least one positive root of this equation. And we have the other 3 compartments expressed through this S star. How many roots do we actually have? It depends on what happens to the other coefficients. So we know for a fact that there will always be at least one, and as I'll show you in a minute, there can be more. What happens when we look at stability? Again, now the system dimension is even higher, so it's a quartic, and we still have inside those terms which hide the exponentials. So if we take just one of those 2 time delays as positive, the other one as 0, the structure of equation does not change, so the analysis actually would carry over and it would be the same in both cases. And again, we can find an equation for the Hawk frequency omega, we can find the critical time delay, and that's what it looks like. So what do I have here? On the vertical axis, and likewise here, that's the steady state value of the mRNA. So that's my m variable. And on the horizontal axis here, that's the number of transients. So that's how many of transgenic copies are produced when mRNA is transcribed. Now, what was B1? So B1, that's the rate at which RNA silencing complex binds to mRNA. So that's effectively how effective RNA interference is, because it tries to destroy mRNA. And initially, so if this number is reasonably small, then we do have, for every value of N1, just a single possible value of mRNA. They are stable for small N1 and for large N1, and somewhere in the middle, they're unstable. But as RNA interference complexes become more and more effective, so this means that the number B1 grows, we get a hysteresis. So effectively, the curve turns, and there is a range of N1 values where we have 3 possible values for M, and correspondingly 3 possible values for S and all the other variables. So the top one, that's where there is no RNA interference. The bottom one, that's the silent state, because that's where RNA interference is working, so the number of, or the amount of that mRNA is very low. That's exactly what we would like to happen. But the point is that they actually coexist in this range. And as the effectiveness of the RNA silencing complex increases further, the range of number of transients for which this happens also increases. So there is a much wider range of values of N1 where we can have a silent state and a non-silent state. We did the same for B2. So B2, that's how we are producing new double-stranded RNA from mRNA. Here the story is somewhat similar, but it goes in the opposite direction, in that the more effective we are at reducing mRNA, the hysteresis curve, which initially had quite a wide range of values, becomes more and more narrow until eventually it completely disappears. So in this case, for every value of N1, we just have a single value of the associated mRNA. Here we were in a bit of a pickle. Why those names? Because when we, for example, start here, it's clear that we are just dealing with a single value. It doesn't matter what we call it. But then it's exactly the same curve that changes its shape, and then we would still like to have some correspondence between them. So for the purposes of being able to distinguish between different steady states, E1, that's the silent state, so that's the lower one. E2, when it exists, is the one in the middle, and E3, that's the one which has the high value of mRNA. Obviously, when we go all the way to the left, we only have E3. When we go all the way to the right, we only have E1. And that's the same story for another parameter. The only difference here, so what is this? That's the rate at which double-stranded RNA is produced from the garbage and the mRNA. And again, as it increases, the numbers on the vertical axis decrease, so the steady-state concentration of mRNA goes down. But unlike the previous two examples, the nature of the curve does not change. So we always have a hysteresis. There will always be some range of the number of transgenic copies where we will have a silent state and the one where RNA interference is not working. This is the picture which parts of it we could get analytically, the rest we had to do numerically. So what we are doing is we are looking at our steady states, be it E1 or E3, so either the silent state or the one where RNA interference is not working. And we look at how the time delay and in particular time delay tau2 affects stability. And what we see is that, so that's the range between here and here, where there are two, where we have all three steady states present. So the one in the middle, E2, is always unstable. The question is the upper and the bottom one, so the silenced one and the one for which RNA is not working, what happens to them. So E1, that's the silenced one. And what we see is that for sufficiently low number of transgenes, if we increase the time delay tau 2, it can become unstable. Same happens for the state where there is no RNA interference. But actually, this transition to instability, it happens for much higher values of the number of transgenes. And here's how it actually looks like. So we have our hysteretic curve. The shape of the curve is completely unchanged depending on the time delay, because the steady state values, they do not depend on time delays. On the other hand, stability changes, and it changes qualitatively. Let's have a look. We start with just the original curve with the hysteresis, and then when time delays are increased, the silent state, it loses its stability. So whilst it was stable everywhere here, now there is a part of this curve where it exists, but it's unstable. So one could expect a periodic solution around it. And something similar happens to the state which we have at the top. That's where RNA interference is not working. Again, if time delays are increased sufficiently high, then this state can also be unstable. So at least in principle, there could be a potential for periodic solution there. And what I have here is all parameters are fixed. The only thing that's different is a choice of initial conditions. So what we see is that in here we have initial amount of double-stranded RNA versus initial amount of garbage RNA. So if there are no time delays, then almost every initial condition results in the state with no RNA interference. And the basin of attraction of the state with, which is silenced is very small. If we increase the number of transgenes, then the situation changes. And as we go from here to here, which means that we increase time delay, and the same goes here, what is resulting is an increase in the basin of attraction of the silent state. And for high number of transgenes, if you compare the axis, the vertical axis in those two figures, it becomes clear that the basin of attraction of the non-silent state also increases. What's most interesting is this picture. That's the regime where we have a coexistence between a stable non-silent state and a periodic oscillation around the silent state. So in this case, depending on where we start in terms of, say, initial double-stranded RNA, if there is enough of it, then we will have not stable state, which is the silenced one, but rather oscillations around the state, if the time delays are reasonably high. And that's what it looks like in actual numerical simulations. So these two figures, they have exactly the same values of parameters. The only thing that's different between them is the initial conditions. Either we go to a stable, steady state which is not silent, so that's where RNA interference is not working, or we go to stable periodic oscillations around the silent state. And those two coexist. So whether the plant will exhibit this phenotype or that phenotype, or if we talk about mammalian cells or any other organism, in a sense, it also it effectively tells us that actually it depends on what is the so the initial conditions play a very important role. And in a second, I'll explain in practical applications why that's important. And the last figure, this is where we go to a stable silent state. But what one should observe is that for a very long period of time, we actually have high amplitude oscillations around the state. Why it's important is because if we compare this to standard models of immune response, again, in clinical practice, we can't argue things like, well, if we wait long enough, so in the limit of time going to infinity, or if we take an infinitely large population, because when we have a patient in a clinic, we have to tell them something. And one of the arguments is that, say, with immune response, what we have as relapses and remissions in autoimmune diseases, these can be interpreted as endogenous oscillations of this kind, where after initial challenge with a pathogen, in many cases it's a virus, immune system tries to effectively build response against that viral challenge, but when it clears it, it then takes the system into a regime of sustained oscillations, where even if there are no any other pathogens around, then the person would still experience those relapses and remissions. But for that, they don't need any exogenous factors. It's all intrinsic. So I hope I've convinced you that at least it's interesting and it's actually one of very fundamental processes that takes place in all our cells day in, day out. For some organisms like plants, it plays a very profound role and very fundamental because it's one of the main mechanisms of how they fight off viral infections. And even more importantly, I hope I have at least given an illustration why time delays that are purely biologically motivated, again, they can have really a significant effect on the type of dynamics that such systems do exhibit. Some of the things that would make sense to consider, but were not done here for the purpose, purely for the reason that you can't do everything at once and will have finite lifetime. We did not include spatial propagation of the warning signal. Again, that's something that can be done in a reasonably straightforward manner. Effectively, it would just be a modification of the system that would include the spatial component. We would probably have partial differential equations with time delays. There would be mathematical challenges of how to model that and how to simulate them, but definitely it would be a very faithful representation of biological reality. Also, when we talked about plant viruses, I never explicitly included the viral population. We just said that there are susceptible cells that interact with infected cells and become infected therefore. But actually, one could explicitly have population of virus particles that are spewed out from those infected cells. Then they travel the distance and after a period of time get inside susceptible cells. Again, there are definitely very clear ways to model that. It wouldn't be too much of a problem. One could also look at virus life cycle, because again, even for a single cell, all those parts of the virus life cycle, they have non-negligible times. It takes time for viral cell to get inside the, so for virus particle to get inside the cell. It takes a period of time for that virus particle to change genetic machinery inside the cell. It takes another period of time for already infected cell to start producing new virus particles. All of those would introduce additional time delays that can be explicitly modeled. And I did no argument for how we take account of coevolutionary dynamics. Because again, in here, it's assumed that everything is static, so cells always behave in a particular way and their viruses behave in a particular way. But as we know, quite recently, not only do they co-evolve, but they actually do this on a reasonably short time scale, which is very unfortunate for us. Because now it's argued that perhaps the biggest challenge to humanity in 20 years' time, it will be antibiotic resistance. So we can do as much as we like and really go to another planet and be very advanced in lots of our endeavors. But such a simple thing as just being able to fight bacteria, well, we are already struggling. So understanding coevolutionary dynamics, again, would be really of great benefit here. And just to close this off, why this actually is important. So RNA interference, because it's such specific for individual genes, it's perceived one of the really very promising strategies for all the different kinds of medical treatments. In particular for cancers where there are definite genetic components. There are already several trials now ongoing that are based on using the mechanics and the exact mechanisms of RNA interference for treating those diseases. Cancer is one thing, but actually lots of infectious diseases, again, some of them just turn up like Zika, which we never heard about until last year. Others that have been around for a while, but again, since it's a treatment that's very specific, it only affects expression of particular genes. Again, it holds a lot of promise. Same goes for macular degeneration, where again the trials are now ongoing. So that's an age-related condition where with age the vision becomes blurred. But also there are applications to biotechnology, where in biological experiments it's perhaps one of the most widespread and most efficient techniques for controlling gene expression, because we want to, if we want to have a robust handle on expression of individual genes, that's exactly what we will do. In food industry, again, now all the stuff with genetically modified plants, where we can design them in a way that would be much better for human health, or if we say want to increase consumption, or if we want to design plants that would be able to resist viral infections. Actually, University of Surrey, where I did my PhD a while ago, now they have a chancellor who is an Australian, and he is involved in a project. Just a couple of months ago, they released a new protocol where they have helicopters that are flying over fields and spraying them. They're spraying them with substance that, through the mechanism of RNA interference, induces resistance to particular viruses. So it's already been tested. It's been rolled out, and for that reason, it's also a very promising thing. And the last example is what's known as an Arctic apple. That's a real thing that's on the market. It was approved by FDA, I think, a year ago. What they did is they knocked out a gene that is responsible for polyphenol oxidase. Effectively, it's a non-Brownian apple, which is very good if you have to transport them for a long distance. So in the UK, where the weather is not as great as, say, here, if you transport apples and some of them are accidentally hit or if it's squashed during transportation, they just do not brown. So they remain at least visibly the way they were initially there, just because the gene that's responsible for producing that extra color is completely knocked out. Thank you very much.
Thanks a lot for the fascinating insight into the dynamics of RNA interference. I'm sure there are more questions.
Messenger RNA molecules transcribed by proteins are known to be in quite small numbers inside cells. My question is, in this case, they are known to be very noisy, and to vary the numbers of these molecules, messenger RNA, from cell to cell that are clones. So my question is how your study, would, the conclusion would change, if you allow for this kind of stochastic effects, intrinsic noise, all these things.
Good question.
Well, and in some of your figures, in the silencing part, for instance, have quite small numbers of RNA molecules. Also, in the oscillations have a part that
the first part is so in the model where we look just at what happens inside the cell. There is no question one could easily try to look at what would be the effect of just noise in that situation, and to what extent the results that I have shown here would be robust with regards to that. That's in some sense at least mathematically that would be a reasonably straightforward thing to do. The other part of it, which is what you say, the significant variation in the amount of mRNA between different cells, that would probably be a much more challenging thing to do, because then I would think that we would have to work not with cells like this, but rather we say that we take a population of cells and a distribution of the amount of mRNA inside them, and then for this distribution, which at least for the distribution we could use experimental data, just to get an idea of the range and just the shape of distribution. And then we could say, right, if we have something like this, to what degree the results that we have here would carry over. Yes, of course, the model would have to be much more involved for that, but and I don't really know what the ultimate answer would be, but I think it's definitely doable and definitely worth pursuing. It's perhaps on par in terms of say physical significance and the next step with explicitly having warning signal. Because again, we know for a fact that there are things that are spreading. Most probably it's the actual mRNA, so it's not the double-stranded RNA that's propagating. And there are experiments, in the UK there's a dedicated institute that is looking at plant science, and they actually have experiments where they are trying to measure exactly that.
More questions?
Is it important the way the plant grows? Like there is a group of stem cells that reproduce and the other don't reproduce. So it will be probably a linear growth more than an exponential.
I think it's not so much the way it grows as a matter of time scales. I think here it's a matter of what are the relations between, say, the speed at which new cells appear to the amount of time it takes for the warning signal to spread to the, all the rates that I have in the model. The inverse of them, they give you the characteristic time scales. So say lambda, which is transmission rate, 1 over lambda has the dimension of time. And like the recovery rate, 1 over that gives you the average duration of infection. So perhaps to answer your question, one would have to say, right, when we look at all those characteristic timescales that are associated with different processes, whether if we look at a different type of growth, it would happen on a vastly different timescale, and then again, maybe it would actually help you simplify the model. Because if your plant grows very slowly compared to everything else, then for all intents and purposes, you could say it's slow timescale, it's almost constant for the purposes of this particular process. Or the other way around, it could be so quick, then actually you would have to rescale everything else and say, right, the most important thing is the growing plant. No reason why you can't include any of them. It's a matter of probably just what is the minimum model that you could get away with that captures the essential biology and physics that you would want.
And this could simplify your spatial problem, because if you take something like a root, becomes a linear growth, one-dimensional, and so you could perhaps simplify your spatial problem, solving like a carrot.
I agree. Actually, there's a colleague in my department who is doing all kinds of cell models on changing domains. So it's effectively a leaf that's growing. And mathematically, you have to have means to describe that. So even with partial differential equations, but geometry that itself is changing in time. So it's not that you have your square or rectangle or something on which you solve your problem. Rather, the geometry itself, according to some rules, is changing. And then on top of it, you run your process, which is exactly what you described. Again, biologically, it would make perfect sense. The only question is what kind of models you would be able to do there that they would give you some information.
I have a typical experimental course. So what chances do you see to close the loop? So going back now from your modeling results to the biological observations. For example, you find quite characteristic states like the stable periodic oscillations. What chances are those to, for example, identify those also in the experiments?
Well, I think ultimately it boils down to what kind of tools and techniques people use and what state of the art is there. Sure. And I think even now, things are getting better, but I don't think one could make any robust predictions. Another part of the work which I didn't talk about here. So for 10 years I've been collaborating with a colleague who is an immunologist, and he runs experiments in a lab. But when we try to do models of autoimmune responses, the problem is that for lots of essential mechanisms, there is still no consensus in the field, because it's impossible to measure, and different groups, they have different perfectly viable explanations for what's happening. And there are just literally no physical tools that currently they could use in biology to unequivocally say it like that or like that. And until we get there, basically we just go with the prevailing opinion or what our intuition or what our current understanding tells us. Of course, we would love to do better. It's a matter of what can be robustly measured that we have faith in, so that we could use those experimental values and plug them into a model. Because until we are confident with that, then it's I'd rather have a general intuition as a physicist and a survey of what are the qualitative types of behavior that we can observe, and then try to see whether actually, as you say, they are implemented in the field, whether these things do occur or not. Because beyond that, I'm afraid I have actually no forecast on how the experimental side of this would develop. We'd love to, but...
Thanks a lot. So if you have more questions, I would like to continue discussing with Kostya. He will be here still this week and next week.
Thank you very much.